Hi, my name is Jeremy, and I'm a research associate at Simon Fraser University. And today we'll be presenting our work on developing a computerized approach to detect pigmented skin lesions from 3D skin images and tracking these lesions across time. So as an outline, we'll note that previous work has reported pretty strong performance using computerized approaches to predict different types of skin disorders using 2D clinical or dermoscopy images. However, there's been less research on computerized analysis of 3D images, especially those taken over time. So that's what we'll be focusing on in this talk is our approach to detect and track lesions on 3D skin images. Now, clinically, this has the potential where it can reduce the amount of human effort that would be required if we wanted to monitor a large number of atypical nevi over time, and this might help improve patient outcomes. So in the past couple of years, there's been several publications now that are reporting that computers are reaching human level diagnosis performance on 2D images, so typically using dermoscopy or clinical images. However, the 3D aspect provides some important context. So for example, if we're imaging a wide area of the skin, we can view multiple or irregular nevi. And there's some evidence that this helps with patient outcomes, as well as if we want to track lesions over time, we can then monitor changes to the lesion. And this might be helpful for um, early melanoma detection. So for this work, what we've proposed is a computational pipeline for whole body longitudinal lesion detection, where we start out, as you can see on the far left, by imaging a subject, so doing 3D imaging. This gives us a 3D colored mesh of the subject. We can then take the surface of that mesh and unwrap it to a 2D image. And then when we have the 2D image, this allows us to apply, say, state-of-the-art machine deep learning approaches or other machine learning approaches to detect the lesions. And then once we've detected these lesions, we can apply our matching algorithms that allows us to match lesions across time or across different scans. So one of the big challenges with this approach is getting data that we can develop our method on, as well as testing our method here. So we've looked at three different sources of data. The first was a handheld scanner where we imaged a mannequin where we had stickers on the mannequin that represent lesions. The second one was a dataset called FOST, which has 3D human subjects but without any texture information. So we added different pose variations, skin tones, as well as synthetic lesions. And then finally, we looked at a data set called 3D body text, and this has 3D human subjects with texture, so with skin. Uh, and the lesions are visible on there on the skin, but were not annotated. So on the left here, we can see the skin of the subject on the two-dimensional image that was mentioned earlier. And what we did is we manually annotated over 17,000 different locations from 200 different subjects. So as an example, you can see on the green boxes. And then we can use these human annotations to then train our deep learning or machine learning um, approaches to detect these lesions. And you can see here we have the green is the human annotator and the red is the AI annotator or the computer annotations. And then given these lesions that were detected on 2D, we can then map back to the 3D body. As you can see here, the red boxes are the lesions that are detected, and we put them back onto the 3D subject. And then we're able to do our analysis on the 3D subjects as well as visualize. And we can also then label lesions that are of interest if we want to track the lesions, as you can see with the different numbers that are on here. And this then we're able then to match the lesions of the same subjects across different scans. So here we can see the same subject in two different poses, and our automated approach that we're developing considers both the body location and the lesion distances to do this matching. In terms of results, when we compare our AI or the computer annotations with a single human annotator, we achieve a fairly high recall, but a lower precision. So what that indicates is that the the AI does a pretty good job at detecting all the locations that the human annotated, but it finds other locations that the human did not annotate. To try and better understand these numbers, we did another experiment where we had three human annotators go through and annotate the same set of images, and we found out that two out of the three human annotators actually agreed more with the AI annotations than they did with any other of the human annotations. So this suggests that the AI annotations or the computer annotations are performing at a similar level to the annotations we get from a human doing the same task. So to conclude, we've presented a promising approach, a computerized approach to detect and track lesions on 3D skin scans. And clinically, the hope is that this could potentially reduce the amount of manual effort that would be needed if we wanted to monitor a large number of suspicious lesions over time. And that's it for the talk. Thank you very much.